already now. And then hopefully we'll be able to share it later on the YouTube channel. So Gregory, over to you. The floor is yours.
Thank you, Salman. So I'm starting with this picture. You may know Dr. Spock, and what he's now having in his hand is the medical instrument that was used normally by the doctor of the crew of Stargate. And this instrument, they allowed the doctor to analyze either a human body or any form of life or mineral. So it's kind of genetic scanner. And if today we would be confronted with aliens looking at us with a genetic scanner like this, what they would see when looking at us, they would see that we have about 410 to the 13 bacteria in our cells, and which is about the same as the number of human cells that we have. Is like slightly less. It's 310 to the 13. So we are composed of about the same number of bacteria than the number of human cells. What he will see also is that our bacteria, so he would see 3.3 million genes coming from the microbiome, and as humans we have 22,000 genes. So looking at the genetic diversity, we have about 150 times more bacteria genes in ourselves than the human genes that are our human species genes. The number of bacteria that you can find in human microbiomes is about 5,000, and it represents about 2 kilos for an adult. And so the alien would classify us as a kind of a bacteria consortium. So we are not an individual, we are an ecosystem. And this, for me, is very similar to for example, this Amazonian forest, which used to be the source of inspiration for the pharma industry in the 20th century. Most of the chemical, active pharmaceutical ingredient coming from plants, from and the minerals, they're all coming from the nature. And because of the diversity, the Amazonian forest was one of the great sources, but maybe in the future, the Amazon rainforest for the 21st century will be our microbiome, which is very close to us. And if you really look at like substances like poisons that have been used at different concentrations for drug purpose, as pharmacologic agents, they are always there because of a reason. If you had a plant with a poison, it's because this plant didn't want it to be eaten by certain animals. So it's all in the communication between the flora and the animals that where the drugs where identified the drugs. So there are a number of communication pathways between our microbiome and ourselves, and understanding these will certainly lead to a number of future development in the pipelines of the pharma industry. We're not totally there yet, but in this presentation I will cover how I see the opportunities that the microbiome will bring to the healthcare industry. So not only the pharma, but maybe also the cosmetic, the food supplements. So in general, the healthcare industry. When we talk about microbiome, many people maybe believe here gut microbiome, but we have a number of different microbiomes. The hair as a microbiome, the nostril, the oral cavity, the esophagus, the skin, very important, the stomach with, for example, Helicobacter pylori, which was one of the importantly identified bacteria there, and the vagina, and so the colon is in the gut microbiome, is most of the gut microbiome, which is the most important microbiome we have, but they are all important, and many companies involved in cosmetics, for example, are studying and working on the skin microbiome. A very difficult question is, what is a healthy microbiome? And some people believe that you have a standard microbiome that is healthy, and everything that is changing from this is a disease situation. This is more complex. A microbiome is an ecosystem, and it's an equilibrium. And you have some equilibrium states there in the valleys in this graph, which are healthy. And after a perturbation, it could be an antibiotic treatment, for example, you're moving from disequilibriums to a loss of equilibrium, and you can go to maybe another valley, a metastable situation where you are not in a healthy microbiome state, or after a resilience, go to a different composition of the microbiome than you had at the initial stage, but that is also healthy. So at least the consensus now is that a healthy microbiome is something that is stable and that has a large diversity. Typically, it's not bad to have some pathogenic strains. If you have some salmonella, if you have some enterobacteria at some levels, you are in some way protected from an infection compared to someone that never has been confronted where to this kind of bacteria, pathogenic bacteria, and will be perturbed and move to a disease state if confronted for the first time. What has really created a boom in the microbiome science is the conjunction of different techniques that became available. One is the technique allowing us to sequence the microbiome. And I remember I remember that the first time we sequenced the human microbiome, it was breaking news. I was probably a student at the time, or just now we are able to sequence a full microbiome of an individual in a few hours. So getting these million genes and we're able to reconstitute whole the qualitative and quantitative composition of an individual microbiome. You need about a DVD to handle this number of data. So it's a huge amount of data. And if you want to be able to analyze this data, the conventional statistics will not help. And a computer like the computer we had 20 years ago will not also be able to handle this amount of data. So it's also the progress in the capability of storing data, as well as the computer chips that are also able to make more calculation, and the arrival of the data science, you can call it artificial intelligence, that allowed us to store the data from the metagenomic analysis and start to work with this data and analyze cohorts of data. You have a number of cohorts that were a difference places in the world in this science. And that's really only possible today because of yes, sorry?
No, keep going, please.
Okay. Only possible today because of the capacity of the computers and also the possibility that we have to do this metagenomic analysis. So I have done this very simple schematic to really help to show what is my mind map and how I see there's opportunity offered by the microbiome. I have to say that the names that I'm using here, prebiotic, probiotic, postbiotic, and antibiotic, are not exactly in the official definition, especially probiotic. There are some official definitions now. So I am just using this slightly differently from their official definition, just because you see pre and post, anti and pro. So it's oversimplification, but it's a good way to see the different approaches that could be used to get advantages from the microbiome. There are two other red boxes that are not changing the microbiome, but that are also offering opportunities for the healthcare industry. One is the diagnostic, analyzing somebody's microbiome just to predict either susceptibility to a disease or to detect an ongoing disease is a possibility. And the other is also analyzing the microbiome as part of the preclinical part of the development of the active pharmaceutical ingredients developed by the pharma industry, because this is becoming a concern now. Does a drug modify, alter the microbiome? And it's an important point to assess now, and which is kind of rather new in the different steps of the development of a drug. So you have a microbiome, let's say that's the gut microbiome, and there might be good bacteria, bad bacteria, and you want to have a healthy microbiome or to cure a disease. The first and most obvious approach is the prebiotic approach. And I would say, and that's why it's broader than this term, nutrition. Here, I consider it as a part of the prebiotic approach. The way you eat, the diversity of the food that you take will definitely have an influence. Somebody eating only veggie will have a very different microbiome from somebody who's eating only meat, and the type of food and the diversity is very important. So the real definition of a prebiotic is really ingredients, normally not food, that will serve as food for bacteria, and in some cases may influence some species compared to other species. So that's very important. You will find these prebiotics as ingredients for food supplements, but we never emphasize too much how much nutrition and an equilibrated and diversified food is important for the microbiome. The very oldest way to influence the microbiome, and it's not always positive, is the antibiotics. So we don't do really this on purpose, but when you take antibiotics, you can really alter your microbiome. And that was known from long time ago, and people also used to take some strains, and including yeast like Saccharomyces boulardii after antibiotic treatment, or some people would recommend yogurt. because it's known that we need to reconstitute our microbiome. Antibiotics, it's interesting to cite this here because a successful antibiotic for the pharma industry is a broad spectrum antibiotic that is absorbed by oral route. If you add exactly the opposite, it probably becomes a tool to have some specific intervention on the microbiome. A very narrow spectrum, not absorbed orally, could be kind of a surgical tool to remove some strains that we don't want to have in the microbiome. So some people call this contrabiotic, so it's kind of a specific antibiotic. But the most specific antibiotic, if I may say, are the bacteriophages, which are viruses targeting some bacteria on a very, very specific manner at the strain level. So bacteriophages are probably going to be interesting tools to modify the microbiome and remove some strains that are not wanted. There have been also some, for example, we are talking about gut microbiome, but just as an example, there is a company that has done a clinical trial with a cocktail of bacteriophages in burned people, so to prevent septicemia after a large burn of the skin. So I have these three approaches. I put them in the antibiotic section. So the exact opposite is to bring strains. So we here consider potentially removing bad strains. You have a possibility to bring beneficial strains at different levels. And I call this probiotic, but you see how much it's different from the official definition. Doing a fecal microbiota transplant is bringing the whole microbiome from an individual to another individual. So there are a number of companies developing these FMT, fecal microbiota transplant. From there, I mean, it's not always you don't always know exactly what you transplant. In some cases, you also transplant bacteriophages. So it's an ecosystem that you transplant. And there is always also a resilience of the person receiving the transplantation. So at the end, you probably reach another equilibrium. A probiotic consortium, it's also an interesting approach. And it's not a FMT, so you don't give a full microbiota, but you give it's an approach with 5, 10, 15 different strains together that will have a desired result. Another approach is I'm going from the broadest to the most specific, a single strain for a certain purpose, and we discuss the different mechanisms of actions there. And it's not for tomorrow, not so soon on the market, but you could even give a genetically modified probiotic. The industry is using E. coli with plasmids to produce some biological components. You could imagine that you give the bacteria and you do in situ production of these components. There are sometimes some discussion of people opposing a single strain versus consortium, and the kind of arguments are, no, consortium is better because it's already kind of an equilibrium, a collaboration between different bacteria, and a single strain may not change too much the microbiome. This is, I think it's not about what is the most successful approach, it is how is discovered that is leading to either consortium or single strain. if you use the metagenomic information of big cohorts and you start to use data mining and artificial intelligence, you will find correlations, and that will never be on one single strain. You will find that 10 strains have been increased or are increased in the healthy population compared to a certain disease population. And that's how these probiotic consortium are designed. So no understanding of the mechanism of action, just as a result of the data mining and the analysis of the metagenomic cohorts. The single strain is more a bottom-up approach, starting from a mechanism of action that is identified in a strain, and then because you have this mechanism of action and you understand the role of that strain producing that specific component, that you come to a single strain approach. So that's, to me, it's more different approaches, top down and bottom up, rather than one is more successful than the other. The postbiotic approaches, and again, I insist, it's not really the real definition, are anything that are using the result of what is the bacteria doing. So all these, even this probiotic approach, and if you consider single strain, are interesting because they produce some metabolites, they produce some proteins that have an interaction with the host. So in this case, sometimes, and we had recently an example with Akkermansia muciniphila, dead bacteria, they do contain these components of interest, and you may not need a live strain, and it might be successful with a dead bacteria. It's you may say, why dead bacteria if you could do a live bacteria? Sometimes also, and this is the case of Akkermansia muciniphila, it's very difficult to maintain the strain alive, stable. So you better kill it and control the quality of the dead bacteria rather than having something that is live, that is moving quickly and dying quickly. There are immunity modulations, there are metabolites, there are small molecules. This is a little bit different. Sometimes it's taking inspiration from the molecules produced by the bacteria and developing a drug mimicking these, for example, some proteins from the bacteria. So taking the pharmacophore part and only a fragment of the protein produced by the bacteria. And sometimes chemically modifying to increase the stability. So this is more taking inspiration from the bacteria. So here are some examples of companies that are involved in these fields. Here you have a lot of people producing prebiotic ingredients. Eligo Bioscience is working with bacteriophages. I mentioned this clinical trial on burned people with the bacteriophage combination is Ferricides Pharma. Biomix is also a bacteriophage company. Enterum is a small molecule company. TargEDys is doing small molecules, but also probiotics. Amansia is dead bacteria. Day 2 here is personalized medicine. This is an Israeli company that has used the Weizmann Institute cohort to measure how people do insulin how people have glycemic peaks depending on what they eat, and they found that the way that the glycemia peaks depends on the type of microbiome they have. So they are making, so their service is to propose to do a metagenomic analysis of their own microbiome, and then these people receive a personalized recommendation of what kind of food they could eat to avoid glycemia peaks. Sayota Therapeutics is working on strain and strain consortium for allergy and asthma. Pharma is a FMT company. IBT is also a single strain company. Vedanta has a consortium of about 15 strains. Isopia is also a single strain company. So these are examples. And another thing to consider, it's quite interesting, is the different regulatory categories. You have food, you have drugs, you have drugs, and in this case, you have both food and drugs. A live bacteria can be a probiotic as a food supplement or a live biotherapeutic as a drug. I'm accelerating slightly. I want to go into one example. I mentioned TargEDys as a probiotic single strain company, and I will, as an example, show how the mechanism of action of single strain probiotics can be very, very close to pharmacology. The molecular mimicry concept that we are using is identifying sequences in bacteria genome that are mimicking the sequence of human hormones. Here is an example of alpha MSH. I'm sorry, the lines here are not totally aligned, but you have the mimicry, and the pharmacophore of the alpha MSH is RW, arginine tryptophan, and it's an hairpin that is formed due to the glutamic acid that is E here. So we have a very similar sequence in a big protein, CLPB, here from the enterobacteria that has the pharmacophore and the glutamic acid, so the hairpin, and this is here, the red part of this big protein. And this big protein and its fragments, when it's partially degraded, are able to activate the MCR receptor, melanocortin receptor of the satiety hormone. In progress. We've done the same with oxytocin, doing sequence alignment and identifying different proteins from different bacteria that are able to activate the receptor of the human oxytocin. So let's focus a little bit on the molecular mimicry of alpha MSH, which is the satiety hormone. This is a figure from a Nature review by one of our founders, Sergei Fetisov, which is showing the role of the microbiome in the regulation of the appetite. When people eat food, these foods start to be digested, and the bacteria feed on our food even before us, before it's absorbed. They start to grow exponentially. And in this case, at the moment of the stationary phase, they start to produce the CLPB, which is the mimetic of the alpha MSH. This is activating through the enteroendocrine cell, the release of certain hormones going to the brain, like PYY or GLP-1. In some cases, and sometimes in disease cases, such as anorexia, the CLPB itself can be systemically absorbed, CLPB or fragments, and this is only in the case of gut permeability. And if this is systemically absorbed, you have a strong effect on increasing satiety at the brain level. So this is a physiological mechanism involving the microbiome, and in this case, through this mechanism involving the CLPB protein, which is a mimetic of the alpha MSH. So what we have done, we have developed a strain that is able to produce this CLPB. It's a non-pathogenic enterobacteria, because we didn't want to use pathogenic E. coli or Yersinia pestis or Salmonella. They are all producing CLPB. So Afnialvi is a cheese bacteria. And we have done an extensive preclinical work. I'm not going to describe fully here, but you have the reference, and the presentation will be shared, showing that by giving this live bacteria by oral gavage, we could increase the satiety. We measure in animal the reduce of the food intake, and this has an effect, a beneficial effect on the body weight, as well as the glycemia and even the insulin resistance. Based on this, we've moved to a clinical trial that is has been done as a double-blind, multicentric, placebo-controlled clinical trial in 230 overweight adults. The design is that both groups, the placebo and the treated group, have received a hypocaloric restriction, so minus 20% for 12 weeks. And we measured the effect of the placebo and the AFNI-LVE given orally. Bad news for those following a diet. Our primary endpoint is how many people lose 3% of their body weight at 3 months with 20% hypocaloric diet. It's only 40% of the placebo group with the diet only that has lost 3% at 3 months. It is increased to 57.7% in the case of the group receiving AFNI-LVE. So it's a better result for a diet if you provide satiety, basically. With the other endpoint that was how many people lose 4%. Obviously, you lose some of them, only 30% of the placebo and 46% of the treated group. This has a benefit, is also inducing a benefit on the glycemia. Also have to emphasize that at baseline, everybody is normal. So it's a normal glycemia. We don't have any disease people in this population. They just are overweight. I did not mention that they are overweight between a BMI of 25 and 29.9. So they are not obese. They don't have hyperglycemia, neither hypercholesterolemia. There is also an effect of the cholesterol on the cholesterol. And what is interesting to see is that the placebo group, so if you are on a hypocaloric diet, you are likely to be hungry. So we have asked the people to rate their fullness throughout the study. And obviously here, the gray line is at baseline, 4 weeks, 8 weeks, 12 weeks. They remain hungry, slightly below the middle of the scale in terms of fullness, while we see an increase of the fullness in the treated group. AFNI-LVE is playing on the satiety. It's becoming significantly different from the baseline at 2 months, and it's different from baseline and from the other group, very significantly different at 3 months. It's like 55 millimeters, so slightly above the middle of the scale. But if you are with a hypocaloric diet, you are targeting maybe 50 is good enough. I mean, if you are neutral in terms of fullness, that's already a good achievement despite the diet. Diet plus fullness is leading to satisfaction. We see there is a big difference. The treated group is about 68% of the subjects that are either very happy or happy with the product. 5% are not happy at all. In the placebo group, we have only we have inverted graphs here for very good and good. It's 52% that are either very happy or happy, but 14% are really considering rating the placebo as poor. So it shows that it's a perception also of the effect in terms of satiety of the product that is reflected by this assessment. I'm going very quickly just to say that when we have identified these mimetics of the oxytocin, we also have now a proof of concept. This is a model that is used normally by the pharma industry for antidepressant or anxiolytic type of products. And we have a difference in different tests. So animals that have been receiving chronic stress and undergo some tests like OMACE, where you see that the treated animals have a more are moving in the maze in the open and closed parts, so they have a better exploration which reflects that they have less stress. The tail suspension test is also, you see, accumulated energy to try to claim if suspended is important in the treated, more important than in the placebo group. For swim test, water wheel, and phenotyper. So just to show here that these probiotic approach, and even in this case, as food supplements, they in some case they have a well-described molecular mechanism of action that is also reflected by efficacy in relevant preclinical models and in clinical models like we did with the AFNI-LV clinical trial. So that's the end of my talk. You have my contact information here. And I did not look at it, but I see that we have in the chat some maybe already question. I don't know, not maybe question. Okay.
Thank you so much, Gregory. That was fascinating. And obviously, when one starts talking about this phenomenon, then 30 minutes, 45 minutes is not enough. One can go on, and especially with your knowledge, one can go on talking about it for a long time. My very quick question is, this looks almost too good to be true. Is this some kind of a silver bullet that we're talking about with the kind of research that you guys have been doing for such a long time? And the results, by the way, are very impressive. So is this something which, we can say probably is going to then, in many ways, we compare that, especially with existing treatments like orlistat and everything, seems to have some benefits. So do you think that this could be the start of a new era of treatment?
I hope so, but when you say so good to be true, it's not really. I think the weight management is really a very difficult target. You see these results of the diet, and the results we have. Drugs. I mean, for example, orlistat, which is pancreatic lipase inhibitor that prevents the absorption of the fat. So you may say, oh, that's a fantastic approach. This should be very successful. But if you look at the endpoint, how many people lose 5% of their body weight at 6 months, it's only about 50% compared to 40% in the placebo group. So it's a multifactorial problem. There is not one approach for everybody. For example, this AFNI-LV probiotic approach, we already know that it works on about 60 to 70% of the people. The rest, so the remaining 30 to 40, we don't know why it does not work. It doesn't seem to be microbiome related. It might be psychologic. It might be some people that do not care at all about their feeling of fullness and still eat even if they feel full because they have anxiety. So we have to be humble. I think this is another stone that is a progress. Definitely, I'm convinced that it's better to help people to change their eating behavior than trying to prevent the absorption of the nutrients. Because this is going to reinforce the bad eating behavior. So it's a vicious circle then. And that's what we see in the people doing yo-yo. I mean, they are having a diet, they try something and they stop and then they gain back more weight than what they had before. So it's very important also to consider that these people sometimes and most of the times they also need to be helped by a dietitian, nutritionist, and it's a tool as part of the different tools.
Thank you. I'll stop the recording here.